The study delves into the genetic relationships among populations in China, questioning the out-of-Africa hypothesis with genetic evidence. It examines 28 populations, highlighting a clear distinction between southern and northern groups, with the northern populations being biphyletic. Despite linguistic diversity, significant gene flow exists between these populations, suggesting no independent origin of Homo sapiens in China. Microsatellites were used to construct phylogenies, indicating that East Asian ancestors likely entered from Southeast Asia. This research is part of the Chinese Human Genome Diversity Project, employing advanced genetic analysis methods to explore the peopling of China, Siberia, and America. Using the DSW program by TOTA for phylogeny reconstruction, with bootstrap values from 500 replications, the study rooted its analysis with African population lineage. FIG-1A, which includes 30 microsatellites from 8 Chinese populations, shows a distinct separation between southern and northern populations. FIG-1B, incorporating 15 microsatellites and adding 16 more Chinese populations, reveals a less clear distinction, but still indicates a common ancestry in East Asia. Northern populations form 2 clusters, one with Altaic language speakers and another with populations from Western China. Southern populations form 3 clusters, one of which reflects the origin of Taiwanese Aborigines. The approach is validated by comparing with Bocock et al.'s results using different markers. The paper includes a table detailing the sampled Chinese populations, their locations, and language families. Polynesians from Southeast Asia and the Ami, a Taiwanese Aboriginal population, cluster with Southern Han Chinese in the US to form Cluster S1. 3 Daik populations, Li, Dong, Yao from Jingxiu, 2 Hmong-Mien populations, Shi, Yao-speaking Punu, Cambodians, Yi, and Han from Henan, form the southern group Cluster S2. The second northern lineage, Cluster N2, includes mostly Western populations derived from the southern group, excluding Iwenki. Jingpo and Hua constitute the third southern lineage, Cluster S3. Phylogenies are constructed using the neighbor joining method based on microsatellites, with bootstrap values from 500 replications. This information is derived from Chu et al.'s study on East Asian population phylogeny in the Proceedings of the National Academy of Sciences of the USA, 1998. The study uses microsatellite markers to identify 2 main groups in East Asia, a northern group with clusters Nun and Nitu, and a southern group with clusters Son, Stibuo, and S3. The low bootstrap values for major clusters indicate weak support for the relationships among them. The genetic distance measure DC and the neighbor joining method were preferred for constructing phylogenies due to better performance in the presence of admixture. The study concludes that East Asian populations likely originated from a single lineage, with the majority of the gene pool in East Asia contributed by modern humans from Africa. It suggests that microsatellites may have limited resolution for closely related populations, and that a larger number of loci may not improve this. While focusing on major evolutionary patterns, haplotype-based analysis might reveal minor contributions from an independent origin of modern humans in East Asia. Northern populations in East Asia are divided into 2 groups with weak statistical support, one showing genetic admixture with southern populations, particularly in southwestern China. The study used neighbor joining phylogeny reconstruction and microsatellites, introducing complexity due to genetic distance measures. Linguistic boundaries across 6 language families were often crossed, with southern populations showing pronounced clustering by geographic region, consistent with Chinese population migration history. Genetic evidence suggests southern East Asian populations may have originated from Southeast Asia, with northern populations influenced by Altaic populations from the north. The migration routes of Altaic populations to Northeast Asia remain unclear. Cranial and dental analyses link Siberians closer to northern Chinese and Mongolians than Europeans, and Arctic peoples to American Indians through Beringia. The study concludes that Altaic-speaking populations likely originated from an East Asian population derived from Southeast Asia, with admixture from Mid-Asia and Europe. The findings are based on minority populations, and a broader study is needed to estimate contributions from each source. The research was supported by various foundations and individuals, with informed consent obtained for newly collected Chinese samples. References include works by Underhill, Ishida, Königsberg, Alexeev, Hahn, Dodo, Ossenberg, Turner, Schur, Taroni, and Merriweather, covering topics such as genome research, prehistoric dispersals, craniometry, and mitochondrial DNA in populations, published between 1984 and 1997.